So in this lecture, I want to cover some of the biochemical pathways that the book talks about that help us understand what happens to fats, particularly triacylglycerides or triacylglycerols, excuse me, or glycerides is better. And what happens to them and how they're kind of broken down. And then we can look at how they're built up in future lectures. Okay, so the first one we want to look at is the digestion, which is really the hydrolysis of fatty acids. Now, this is a reaction that is very reminiscent of things we've looked at in the chemistry of esters. That is where we take our triacylglyceride. If you recall, we've got a glycerol backbone. A glycerol backbone that's been reacted with a few esters. So we could have all of the R groups the same, or they all three could be different. Now, what happens is that in the catalyzed by an enzyme, we're going to have water come in plus an enzyme to actually hydrolyze or crack open those esters to release aspects of glycerol. So in the figure in the book, they show that the top two esters break to give us a carboxylic acid with R plus a carboxylic acid with R prime. Now recall that these carboxylic acids are fatty acids. Okay, now when we break down two, it makes it easier for us to transport around the monoacylglyceride and the fatty acids around. It helps with emulsification. These things are just so fatty and insoluble, I guess, lipid-like, a better phrase, that we have trouble transporting them around. So we have to kind of allow enzymes to hydrolyze and then reestablish the acyl groups at various points so we can transfer these things around the body. And then there's some bile salts that help with the emulsification the book goes through. It's pretty cool. But anyway, let's look at the mechanism for this ester hydrolysis. By the enzyme that does this, which is pancreatic lipase. And this pancreatic lipase is the same enzyme that hydrolyzes the esters to make the monoacylglyceride for transport of fatty acids, and ultimately the glycerol backbone too. Okay, so what happens is we use kind of a combination of histidine plus serine for hydrolysis. So the enzyme is this complex array of amino acids, and there's a histidine and serine that are pushed close together in the three-dimensional fold of the enzyme, not in the sequence. If you look, this histidine is at position 263 and the serine is at 152. So in sequence space, they are a bit far apart from each other, but they can all fold and wrap around and come together. And then we get this interesting arrangement where the histidine, which has a side chain with this heterocycle, I'm going to put the double bond over on the other side. So there's the imidazole side chain of histidine, and that's going to hydrogen bond to the OH of the serine that is neighboring, not in sequence space, but in physical space due to the three-dimensional fold. So this hydrogen bond, what it does is it's not a deprotonation event. Okay, we can draw curly arrows, sure, for deprotonation, and we'll violate some rules of pKa's. But pKa's kind of give us a sense of where things wish they could lie in the absence of external factors. Here we're pushing everything close together. We're pushing the histidine nitrogen and the serine OH close together in space. We're locking out some water molecules due to hydrophobic effects, and we allow a substrate to come in. All of that could promote a full-on proton transfer event, deprotonation of the alcohol if needed, And here it's going to be very helpful, where that hydrogen bond is going to enhance the nucleophilicity of the lone pair electrons on the serine OH, allowing them to react with just an ester coming into the active site. I'm going to put R prime here as, here's what I'm going to do. I'm going to, well, no, let's just leave it at this, right? The focus is the curly arrows. Okay, so we're going to add to the carbonyl carbon and push the electrons up. We've done this before. Now, if you want, you could kind of show more curly arrows. You could show that the nitrogen over here pushes electrons into this hydrogen bond, allowing for full-on deprotonation, where that negative charge is going to go back into the oxygen to then show us why that serine OH is willing to undergo nucleophilic attack on the carbonyl carbon. All of that is necessary because on their own, OHs from serine aren't able to add into carbonyl. So it's the facilitation by the neighboring imidazole that gets us there. Okay, so then we have to redraw our mini active site again. And what we have here is a tetrahedral intermediate. I'm going to try not to move atoms. Okay. We've got our mini tetrahedral intermediate as the serine is now directly bonded to the what was the fatty acid. Okay. So what's going to happen at this point is the negative charge is going to come down and kick off the OR group. And what we can do is we can allow that RO group to be to go over and grab the hydrogen. Now, because everything is in close proximity, it could be that there's a hydrogen bonding, there's probably a hydrogen bonding interaction that precedes that, connecting this H to that O. And if we already have that connection point, it makes sense as to why those electrons would want to those electrons or electrons in the oxygen would want to grab that hydrogen. Now, furthermore, we should be reminded that we have a positive charge in this nitrogen atom. So as we deprotonate, we're willing to give up that hydrogen on the nitrogen atom because it neutralizes the N+. That then gives rise to this structure and our serine residue. And our serine is now, whoop, I forgot an O, excuse me. Our serine is now bound to the acyl group directly. Okay, well, at this point, we can allow some water molecules in, and the water molecules can hydrogen bond to the imidazole just like the OH did earlier, and that's going to facilitate addition of the oxygen atom to the carbonyl carbon, and we can push it up. Now, that hydrogen bond is necessary to activate the H2O to become a better nucleophile, because on its own, the H2O is probably not as good enough nucleophile. Though what the enzyme could do is kind of strain the serine acyl group, strain that carbonyl ester so that it makes it more reactive. The enzyme can do tricks like that with its conformational changing. Okay, another way of drawing the curly arrows would be to take the electrons from here and then the pi electrons, and then we push electrons through this way. I like to think about it as though we have hydrogen bonding to activate the oxygen, and then once it adds, the imidazole rips off the hydrogen. Either way, we get to the same outcome, which is another and ultimately, it doesn't matter too much about the curly arrows, so much as you don't like violate some huge rules, like some pKa effects are just, it's hard to really understand what's going on in the active site, like how basic and acidic things are, and to just assume we get the exact same outcome as though they were freestanding. But the freestanding stuff serves as a good reference point. Anyway, focusing on the task at hand, we now have a new tetrahedral intermediate as the H2O has added its nucleophilic oxygen to the acyl group to make a new tetrahedral intermediate, and the electrons can come down and kick off the oxygen on the serine residue. And that's one way to draw it. I like to kind of think that there's a hydrogen bonding interaction here. I mean, they're everywhere. Hydrogen bonding interactions are everywhere. So as this comes down, what we're doing is we're actually pulling off the electron on the hydrogen simultaneously. It's already sort of adducted through hydrogen bonding, pushing electrons through this way. So if we do that, we're going to get a histidine back to its normal side chain, plus, it's not really plus, I mean, all these things are just in close proximity to each other, plus a serine. If you look, if it grabs that hydrogen, it's an OH, and this is plus our fatty acid. So it's this combination of histidine and serine in the active site that accomplishes the nucleophilic addition of oxygen to our triacylglyceride, activating it towards hydrolysis ultimately. If you think about it, we acylate the enzyme, and then water comes in and removes that acyl group, that ester group, from the enzyme active site, releasing our enzyme back in. I mean, the enzyme was consumed and then regenerates, definition of catalyst, right? But it was required for that to occur. And sometimes, if you kind of are reading through this, we have an aspartate that facilitates that initial deprotonation. Okay. So I want to look at now is we have these fatty acids. And what we need for the fatty acids to do is to get back onto the ester. Check my notes here. The adenylation of fatty acids is necessary. Yeah, okay, good, good, good. All right, so how do we, the question now is, how do we put fatty acids back on the glycerol? So how do we put the fatty acids back on glycerol? This has to do with a concept called adenylation with ATP. So ATP is going to be like our energy driver for this reaction. But what we want to accomplish is we want to take our fatty acid and react it with glycerol. I'm not going to complicate it. Glycerol can have some R groups on the other oxygens. Any of these oxygens could react, but we want to accomplish this using an enzyme where we reform the ester, because we have to take these things off to transport them to various parts of the body. Then we need to put these acyl groups back on the triacylglyceride to make our animal fats and vegetable oils that we need in various parts of the body. So how do we esterify, or esterification of, how do we accomplish esterification of a fatty acid? And so here we do, I think it's adenylation, but I'm not going to add the term. The process is the exact same, but here's what we do. We first take our carboxylic acid, and I'm going to consider the deprotonated form. So this is our fatty acid as a conjugate base. Hopefully it's not too much of a stretch of imagination to see that that can get deprotonated. Okay, now we're going to react that with ATP. Now I'm going to draw out the critical piece here, which is the triphosphate part. That's the high energy part, the highly reactive part of ATP. So there's adenosine triphosphate. This whole thing is, I'm not going to draw arrows under there. This whole thing is ATP. Okay, now ATP is interesting because what we have to remember is that the diphosphate is a good leaving group. So we can make an oxygen to phosphorus bond with diphosphate, 2 phosphates as the leaving group. Now, where do you want to connect your oxygen atom? Do you want to connect it closer to the adenosine or further away? It depends on the enzyme that's consuming the ATP, or that's facilitating the consumption of ATP. So it seems to be that that there's a magnesium as a cofactor for these enzymes that binds to two of the phosphate oxygen atoms of the triphosphate core. The oxygen will then add to either the, not in this case, but it could add here or here, depending on where that magnesium was at. In this case, we are going to add to the adenosine side of our ATP. Sometimes it's the opposite side, releasing adenosine diphosphate. Here, we're releasing inorganic diphosphate, where it's just diphosphate floating around without something attached to it. So you add here. The authors point out that this is probably an SN2-like mechanism. I think I was doing addition elimination before, but with the phosphorus 5 center, it's a bit awkward. I mean, it's actually possible to just do direct SN2 reaction with concerted loss of the leaving group. Okay, so that's going to sever this bond right here. And what we're going to be left with is an acyl phosphate where we have inorganic diphosphate shown as the leaving group. All right, there's our diphosphate leaving group. And we call this PPI, inorganic diphosphate. Okay, now this is an acyl phosphate. And if you recall, this is more reactive than a thioester. This is actually our equivalent of an acid chloride in aqueous solution, in water, when biology is happening. So this is our acid chloride. And so what we can do is we can take our acyl phosphate. And I really want to call this adenylation, which is what happens when you make polypeptides with the amino acids, but I won't, just in case I'm wrong about the nomenclature. Although adenosine. Okay, anyway, we have an acyl phosphate. So now what happens is the glycerol oxygen, or excuse me, no. Glycerol oxygen is far away at this point. What happens next is we have a sulfur come in from the cofactor CoA. And the sulfur atom is more acidic, so we could have some base deprotonate it. And then electrons on the sulfur, either as lone pair or just the resulting thiolate anion, could add. We're going to come up, come down, and then kick off AMP, or adenosine monophosphate. And that's going to make a thioester. And now we have adenosine monophosphate, or AMP. Okay, so now this is a fatty acyl-CoA, and this is a thioester. So thioesters have sulfur instead of oxygen, and they're much more reactive than the corresponding oxoester, especially towards some of the nucleophiles we'll typically see floating around in biology. And so what happens is there's an enzyme. Just want to see if they give me an enzyme name. Yeah. The enzyme is an acyl transferase, which is going to swing the acyl group, that carbonyl group, around in the biological environment. And so what you see is kind of a thioester exchange where our sulfur from our CoA that's able to kind of float around reacts with a cysteine of an enzyme, which I'll write as cysteine, CYS. Now cysteine is just like serine, it's just got a group hanging off of it. This time it's an SH instead of an OH. And so what we're going to see is kind of a trans-thioesterification where one sulfur replaces another sulfur, and the enzyme environment is going to promote one thioester formation over another, and there's some sort of basic residue. Anyway, we get to a new thioester plus coash, I guess, or written backwards. But anyway, this is coenzyme A as the free thiol that can go around and do other reactions with acyl phosphates. Okay, so cysteine is now bound. The cysteine residue of our acyl transferase is now bound to the thioester. Within the enzyme active site, we can now finally bring on glycerol and take one of the OHs from glycerol and react it with the highly reactive thioester. I'm not going to push arrows for this because this does have some nice important hydrogen bonding things happening within the active site. And it would be a little bit of an oversimplification. Additionally, I want to just remind us that any of the oxygens on glycerol could react. Okay, so we made it. We put the acyl group back on the glycerol. So we have to take it off and put it on. These things aren't just one-to-one reverse. They look, the mechanisms may look very similar, but we use different enzymes in either case, involving thioesters and acyl phosphates to put things back on. It's tough to like make esters, but cracking things open, a little bit easier to do, depending on what we're doing. Okay, one thing I think is interesting, and I just wanted to point out, is that the book has a section about the fate of glycerol. I thought this was super interesting. So if you look at glycerol, gosh, why did I draw that wedge? That wedge is just is a little bit too, is steering you in a little bit of an interesting direction. So if we have glycerol here, this is an achiral molecule, but it's an achiral molecule with, if I put this here and this here, it turns out that that's the same as the mirror image. But if we do this, we can actually label this as our prochiral S OH and our prochiral R on the right. Pro S. That is, if I react there, I actually destroy the bilateral symmetry in the molecule and make glycerol chiral, which is kind of interesting. Okay, so we can use an enzyme that consumes a adenosine triphosphate by letting the adenosine diphosphate be a leaving group. So this is where we're actually reacting at the opposite end of the adenosine chain, and that's going to put, and I just want to make sure I have my stereochemistry right here. Yeah, that's going to put a phosphate group on the right side of glycerol. Okay, so we have the right side of glycerol, and then what we do is we will oxidize this using NAD. And so this is our pyridinium. We have this pyridine with a positive charge. It actually can accept hydrogens and then donate them back. Here it's going to accept the hydrogen. And which hydrogen is it going to accept? It's going to accept the dashed hydrogen and completely destroy the stereocenter. So it turns out we get the R product formed selectively. Okay. Yeah, because we have 1, 2, 3. Yep, it's R. So we have the R product formed selectively, but then we oxidize this by removing the red H. The other H on the OH gets just deprotonated through a standard acid-base reaction. And that gives rise to a ketone here. I'm just going to write P there. Okay, and this is dihydroxy acetone phosphate. And I always think this pathway is just super cool because what happens then is this then engages in isomerase to flip positions of the alcohol and the aldehyde. And now this reacts via aldol reactions in carbohydrate pathways. So you kind of take glycerol, this byproduct of triacylglyceride metabolism, and you do a phosphorylation with ATP where you put a phosphorus group on. This goes through a different mechanism than we saw for activation of the carboxylic acid, then an oxidation with NAD to NADH, pulling off the hydrogen I've highlighted, and then a crazy isomerase where we flip the alcohol position with the carbonyl, and you could kind of, if you want to, I mean, there's just two hydrogens that move over. It's like a keto-enol tautomerization in there that kind of helps with this. It's a pretty simple transformation if we go through the curly arrows, which we will in future lectures. But then we go from the lipid metabolic pathway to the carbohydrate pathway. It's kind of circling around in these various important biochemical pathways, but it's all required, right? We have to connect all these things together to kind of sustain life a little bit. What I want to do, this is I'm getting a little long-winded just talking about the hydrolysis. But the last thing I'll touch on is just the mechanism for glycerol phosphorylation. So, because it touches on an important feature of ATP. Mechanism of glycerol phosphorylation looks like this. What we do is we take glycerol, I'm going to draw this hydrogen up here. Here we're going to use a basic residue, which is actually a carboxylate anion, aspartate. It's 245 of this enzyme. Now, this is an ATP-dependent enzyme because the ATP actually engages, it provides the source of phosphorus for our phosphorylation process. ATP is a common phosphorylating agent. Okay, so now what's happening is our nucleophilic oxygen that receives phosphorus is going to react with the phosphorus at the end or further away from adenosine. And that's just controlled by, again, the enzyme and its positioning of magnesium within the active site. So it's actually going to put the magnesium on this diphosphate end, which activates the terminal phosphorus. So we could have the book has the kind of hydrogen bonding to this OH increases the nucleophilicity of the oxygen on the right. And we can actually consider that it's only this right oxygen. The two are the same by chirality, the two terminal OHs of our glycerol. But in the enzyme active site, we can discriminate and have the right side attack the phosphorus. And through an SN2-like mechanism, by way of the phosphorus 5 center, we can actually displace, or we can add the phosphorus to the nucleophilic oxygen and lose the diphosphate leaving group. Now here, the diphosphate leaving group is also connected to adenosine. And so you get ADP, adenosine diphosphate, being formed as a result of this process. So we now have our glycerol 3 phosphates. And the numbering is always a bit interesting. So they put this like SN in front of it for nomenclature. Don't worry about that. Plus adenosine diphosphate. So the enzyme positioning the magnesium at different points in the ATP will control whether it's the terminal side, just putting a phosphate on the nucleophilic oxygen, or the internal side, facilitating facilitating just the addition of the adenosine plus the phosphate present. Okay, sorry, caught my words there. I was looking at something else. Okay, so that'll do it for this lecture. I want to cover fatty acid oxidation and triacylglyceride catabolism in the next lecture because that's a super interesting process. And then maybe that's sort of it for chapter 3, I guess we'll see.